Okay, so let's continue on the immune system. This is immune 3, I believe. We're going to start with antigen presenting. I think this was we left off, antigen presenting. There are 3 types of cells that are called APCs, which stands for antigen presenting. Dendritic cells, B lymphocytes, and macrophages. Then you also need to know of 2 what are called major histocompatibility complex proteins, MHC proteins, type 1 and type 2. Type 1 is found on every nucleated cell in your body. This means they're not on red blood cells. Type 2, those are the only cells that have type 2 are APCs. Okay. Function. Normal with self-antigens that are tolerated. That is, you mix every cell in your body, except for red blood cells, mixes type 1 with a self-antigen, and this informs the immune system that everything is hunky-dory, everything is good. If they're infected by a virus, we're going to see they put a part of the viral protein with type 1, and then this is a suicide signal telling the immune system to come along and kill me. And so those cells have been labeled for destruction. Either transformed cells that become cancerous or cells infected by a virus. Here's the top part of this figure is formation and docking of MHC proteins in a healthy cell with a self-antigen. That's a bad term, but it's a self-protein that's recognized by the immune system with MHC1, and everything is good. Bottom part is a cell infected by a virus. It chews up part of the viral protein, connects it with MHC1, puts it in its membrane. And this tells the immune system, I'm infected by a virus, please come along and destroy me. So, and the cells that will destroy it are cytotoxic T cells. Okay, type 2. So, displayed with foreign antigens on cell surface. So, these dendritic cells, macrophages, and B lymphocytes, they take in the virus or the bacteria or the foreign organism, they chew it up, they take part of that foreign protein and combine it with MHC type 2 proteins, display that in their surface. And that is to stimulate helper T cells, helper T cells that have come along and recognize that foreign protein, that antigen, and the MHC2, and then it will either stimulate itself or stimulate the APC, in the sense of was going to see in the case of B lymphocytes, the helper T cell stimulates the B cell when it sees that antigen. This slide is the showing of an APC that is chewing up a bacterium cell, proteins in a bacterial cell after phagocytoses it, merges that antigen, bacterial antigen with an MHC type 2 and displays it in the surface, in its membrane. Here are the 2 types of T lymphocytes with T cell receptors with specific foreign proteins. It's either, and then it has a co-receptor. It's either CD4 or CD8. If it's CD4, it's a helper T cell. If it's a CD8, it's a cytotoxic T cell. And CD4s interact specifically with class 2 molecules. CD8s interact specifically with class 1 MHC. Okay, life events of lymphocytes. Formation, activation, effector response. The formation in primary lymphatic structures, which is thymus and bone marrow. Activation in secondary lymphatic structures, usually lymph nodes, lymph nodules, spleen, tonsils, appendix, and then finally an effector response, either the site of an infection, which could be cytotoxic T cells, or throughout the entire body in lymph nodes. So they have to be formed first, activated, and then they do their job. That's the effector response. So here, the top part is the formation of lymphocytes, then the activation of lymphocytes, and then the effector response of lymphocytes. In the top one, we have B cells being made in the bone marrow and maturing in the bone marrow. So think of B as bone marrow. Then we have T lymphocytes being made in the bone marrow, traveling to the thymus, maturing in thymus, and T stands for thymus. And they're called naive immunocompetent, which means they're able to do their job right now, but they're naive. How is it they're naive? Is that they haven't come into contact with the antigen that they're specific for. Then they travel to lymph nodes, to secondary lymphatic structures, lymph nodes, spleen, tonsils, so on. And then they're activated there. In other words, they come into contact with the foreign antigen that they're specific for, and then they do their activation response, the effector response, I should say. And what helper T cells do is secrete interleukins that will activate T lymphocytes and activate B lymphocytes. So toxic T cells will secrete these perforins that make a hole in the foreign cell or the infected cell and allow these granzymes to enter in and chew it up and kill it. B cells, when they're activated, become plasma cells, and they make antibodies. And these antibodies go out through the entire body, and whenever they come into contact with that antigen, they bind to it. And we're going to see later on, we'll get into specific talking about antibodies, we'll see how they actually lead to the death of the cell or the destruction of the foreign particle. Okay, T lymphocytes in their formation. The T receptors tested to see that it binds to MHC1 and does not bind to self-antigens. So we have a selection. They got to go to school. They got a positive response. They can bind MHC. Negative, they don't bind self-proteins. They're self-tolerant. If they flunk that test, they're destroyed by apoptosis, even before you're born. Okay, so immunocompetent but naive after they're formed. The T cells, pre-T cells, will differentiate into helper or cytotoxic T cells, depending upon what co-receptor they have, CD4 or CD8. Same process for B cells, but happens in red bone marrow, not in thymus. And then, as I mentioned, they migrate to secondary lymphatic structures, and B cells only end up in the lymph node and spleen. T T cells can be all over the place. Okay, helper T cell activation. So in order to have any response of any of these cells, you got to activate a helper T cell. They are the crucial cell in your immune system, perhaps the most important one. And it's cruelly true that that happens to be the very cell infected by the HIV. That's why it's so devastating, because it's the most crucial cell in your whole arsenal, immune system arsenal. Okay. Helper T cells have to come into contact with an APC that's presenting the foreign antigen and type MHC type 2 proteins. Then if the helper T cell comes into an APC like that, it secretes interleukin-2, which stimulates itself. It's like an auto stimulation. And you have to have there's two parts to the activation. First activation and second activation. First activation is, hey, we got a foreigner here. Okay, second activation is, are you sure? Because we don't want to make a mistake. After it's activated, but for the second time, clonal selection happens. And that is, they go through mitosis to make a whole clone of these cells that all have the same receptor to the same foreign protein. And you make activated helper T cells and memory helper T cells that will remember this encounter for long periods of time, hopefully your whole life. Okay, so toxic T cell activation contact with a peptide fragment with MHC type 1. Then co-stimulation by helper T cell secretion of interleukin-2. So both these cells have the same T cell receptor for the same foreign protein. One of them has the co-receptor CD4, the T cell has CD8. The CD8, remember, only reacts with MHC type 1. They come into an affected cell or foreign cell. Then helper T cells that come into contact with this same foreign cell, and it secretes interleukin-2. And that causes clonal selection of the activated cytotoxic T cells and memory cytotoxic T cells to go out there and fight and destroy these invaders or these unhealthy cells. Okay, here is the co-stimulation to activate T lymphocytes for clonal selection. First, helper T cells. A naive helper T lymphocyte comes into contact with a APC. This is the first stimulation. And then once that happens, then the helper T cell secretes interleukin-2, which feeds back upon itself, binds membrane receptors on itself. And the second stimulation is the helper T cell stimulating itself. Okay, goes to clonal selection to make a bunch of these identical helper T cells. Other side, the cytotoxic T cell. It comes into contact with an infected cell, displaying MHC class 1 proteins with this foreign antigen, and it binds to it. But then it has to come into contact with an interleukin that's secreted by an activated helper T cell, in this case, the interleukin-2. Just like the one that activated the helper T cell, self-activated the helper T cell, now it has to bind membrane receptors on the activated cytotoxic, so that's the second stimulation. And then this cytotoxic T cell goes through clonal selection, makes a clone of itself, and is ready to go out and fight and kill these cells. Okay, activation of B lymphocytes. B lymphocytes, remember, are APCs. They come into contact with a foreign antigen. They bring it in, they chew it up, they display it in the membrane with MHC type 2. A helper T cell comes along. sees that, recognizes the MHC type 2, recognizes the foreign protein, secretes interleukin-4. Interleukin-4 binds to the B lymphocyte, the second stimulation. The B lymphocyte goes to clonal selection, makes a clone of identical B cells that make gazillions of identical antibodies that become plasma cells, and those antibodies go throughout the entire body looking for this antigen. And then there's a bunch of memory B cells left in the lymph node, and hopefully it'll be there your whole life and provide lifelong immunity from that specific organism. So there's the figure that shows the co-stimulation to activate the B lymphocyte for clonal selection. The first stimulation, they come into contact with the antigen, the antigen present, along comes a helper T cell, recognizes it, binds to it, secretes interleukin-4, binds to the B cell, clonal selection. Okay, lymphocyte recirculation. Lymphocytes circulate among secondary lymphatic structures. The response to the cytotoxic T cells, remember, is a lethal hit of toxic chemicals. Foreign or unhealthy cells, and it secretes these perforins that make a hole in the membrane of the unhealthy cell. It secretes the cytotoxic T cell secretes these granzymes that enter this hole, into the cell through this hole, chews up the cell, and destroys it. So the response of helper T cells it's an enabler. It helps all the other cells. It helps to activate cytotoxic T cells, B cells, NK or natural killer cells, and macrophages. They're all stimulated by helper T cells. So there's the effective response of helper T cells to the left, secretes interleukins. These are cytokines that stimulate other cells. There's a whole bunch of them, but we just need to know interleukin-2 and interleukin-4. The effective response of cytotoxic T cells to the right, the secretion of the perforins, and then the granzymes to completely destroy this unhealthy cell. Cause apoptosis of that abnormal cell. Okay, T cell recall response. Now we're going to get into humoral immunity and talk more about immunoglobulins, which are antibodies, and how they work, what they look like, what their structure is, and how they lead to the death of foreign cells or foreign particles. So I'm going to end it right here and pick up this in the next video lecture. So I'll see you later.